Our next speaker, it's my privilege to introduce our next speaker, who's Michael Combe from Cell Signaling Technologies. Dr. Combe is the CEO and the founder of Cell Signaling Technologies. But in addition, he's also the company's research director, both in function and in his heart. He was the director of the Molecular Neurobiology Lab at Mass General Hospital, and he was an associate professor of neuroscience at Harvard Medical School before he left to pursue this industry. And really, CST grew out of his passion for recognizing the need for new tools in order to study signaling. Cell Signaling Technologies has really pioneered a wide variety of antibody technologies, and particularly implemented their use using phosphoproteomics and other approaches to try to study signaling networks in cancer. Mike has published a number of, I think, very important papers, and the one that I think most recently has really been striking is his laboratory was one of two that identified aberrant phosphorylation sites and aberrant ALK signaling in lung cancer. And that is, I think many of you know, has really changed our appreciation of a small subset of, of a modest-sized subset of lung tumors that can be now addressed using ALK inhibitors. So I'm not sure what he's going to talk about, but I'm sure it's going to be interesting. Mike Combs.
This over here, you think, or just? Thank you, Mike, for that very kind introduction. It's a great pleasure to be able to talk about the other side of CST. I think most of you all probably know us as a company that makes antibodies, but we actually have been over the years building technologies and carrying out research. And I'm going to have the opportunity to tell you about three little short stories today that relate to our cancer biology efforts. So I'm going to talk first about the technologies we've developed. Then I'll tell you how we've applied these to study cell line models of cancer driven by oncogenic receptor tyrosine kinases. And then the third little vignette will be this description of our survey of about 500 tumors that we've been looking at signaling. Okay, so I wanted to start with the future of cancer therapies. And I think we all recognize now from the sequencing efforts that there's enormous heterogeneity in cancers. And the goal is obviously to get the right drug for the right patient. And how are we going to do that? We've really got to, in the future, look at what's going on in the cancer cells. So we need technologies to really, in a very personalized fashion for every patient, look at what's wrong with their cells in terms of the genetics and the signaling pathways. And so I think everyone's aware that there's two technologies that are really changing the face of what's going on. The first is next-gen sequencing, and the elucidation here, work at the Broad and many other cancer centers are really defining the genetic basis for cancer right now. And this is, I think, a great advance. And the other that I'm going to talk about today is really efforts to look at proteins and signaling. And there have been similar advances in the mass spectrometry. We now have these very high mass accuracy instruments that allow us to profile protein modifications in a high throughput way, and now allow us to establish pathways and really begin to put, I think the future is to begin to put the pathways that we can identify with the proteomics into the context of the genetic changes that underlie the formation of the tumors. And this will be the kind of information, I think, in the future that we're really going to need. We need the genetics and we need the pathways to really effectively treat patients. So what we've been trying to do over the last 10 years or so is to build technologies to look broadly across all of these signaling pathways. And this is a poster that we put together with Bob Weinberg for his fantastic book on cancer biology. We're working on actually a new edition of this. This was about 4 or 5 years ago. I think Bob's putting out a second edition very soon. It's a terrific book. Highly recommend it, everybody. But anyways, we've looked at these pathways really one node at a time. up until recently. And now the goal is really to develop technology to look broadly across all of these pathways in one fell swoop, identify the pathways that are activated in a diseased tissue, be able to drill down on those and really look at the nodes that are broken and causing the problem. And this will be very informative for cancer therapies. So I'll tell you how the approach that we have taken, and that is to take that complex signaling space that I just showed you, break it up into the different kinds of modifications that are being transmitted through these different signaling pathways. And the approach is to use antibodies that recognize these different modifications, for example, in a very broad way, so that we can look at all of the changes in that signaling space. So for example, phosphotyrosine antibodies allows us to look at all the tyrosine phosphorylated proteins. And we've developed a collection of serine-specific antibodies that allow us to look at very specific areas, and we're trying to cover all the serine-threonine kinases in that space. Similarly, we're developing antibodies that look at acetylation and methylation with the idea that we can broadly profile the signaling that's going on. And the approach that we use is shown here. And the idea is to take a complex mixture of proteins from, say, a cell line or a tumor, and be able to analyze the modifications that are occurring on those proteins. Those are marks that we can read out that reflect the state of the signaling that's ongoing in the cell. And we want to be able to read that out in multiple dimensions. So the approach that we've taken is really to take these samples, trypsinize them, purify out the different classes of protein modifications. For example, using a phosphotyrosine antibody shown here, we can purify all the phosphotyrosine-containing peptides, run them into a mass spectrometer, and figure out what the proteins are, and put together pathways based on that information. And we can do this sequentially with other antibodies that recognize serine, threonine, acetylation, and methylation. And so from a single sample, we can get a tremendous amount of information. This just shows kind of the coverage of the human kinome. There's 500 kinases. Up on the top are the tyrosine kinases, so we can cover that space with the phosphotyrosine antibody. And then these other branches, we've developed motif antibodies that allow us to look at the substrates of all of these kinases. And I think this is a great approach to understand the biology of these enzymes. There's many of these we really have little, if any, idea what they do. Probably the most important, though, view is the enzymes themselves. How do we see which enzymes are actually activated? And to do that, we have developed a new class of antibodies that recognize the activation loop of the serine threonine kinases and also tyrosine kinases. And this just shows a little model of SARC and phosphorylation, the activation loop of most kinases converts it from an on to an off state. And so we've developed antibodies that recognize the phosphorylated activation loop. And these are great markers for kinase activity. And by mixing a number of these motif antibodies that recognize the phosphorylated form of the ACT loop together, we can actually survey hundreds of kinases at once. And so we can look at all of these kinases. And this is a very valuable view, and I'll go into that in a minute. So we can look at the tyrosine kinases, the serine threonine kinases, and similarly we can look in these other signaling areas like acetylation and lysine methylation and also arginine methylation, ubiquination. And we're pursuing that in a number of different studies now. And let me now switch and talk about from the technology to how we're applying that to study cancer cell lines. And we've chosen to study cell lines that represent models of single agent therapies. So we're looking at cell lines that overexpress receptor tyrosine kinases with the idea of trying to characterize the signaling downstream of those receptor tyrosine kinases in such a way that we can map out these oncogenic pathways and identify new targets. And this is important because of the limitation of single agent therapies. We'd also like to be able to understand who is going to respond to these patients, to these particular therapies. And this, I think, illustrates the problem. This is a 38-year-old man with BRAF mutant melanoma shown on the left. It's obviously advanced disease. And then the center panel shows 15 weeks of treatment with a pathway inhibitor, the BRAF pathway. And you can see the incredible effect of these single-agent therapies. But the third panel shows the patient 23 weeks after therapy. And the fact that in late-stage disease, the cells, the cancer cells are able to genetically rewire themselves around the drug target. And so I think what this is telling us, to have durable responses, we're certainly on the right path. We're going to need multiple therapies. We're going to need combination therapies. We need to understand the pathways well enough to hit them at multiple levels. And so that's what we've been trying to do, is to characterize the signaling further downstream of these kinases. And I'm going to tell you about, I'm going to give you 3 examples now of our analysis downstream of these receptor tyrosine kinases in cell lines that are driven by them. So the cell lines, if you give an inhibitor, they'll die. They're completely dependent on the receptor tyrosine kinase, the models of oncogene addiction. And we're going to look at in the tyrosine, serine, and acetylation space with the idea of trying to identify potentially new targets. And this is work that we published. Eileen Gao was a lead author on this. We published this a few years back, but it just shows how if you give one of these cell lines, in this case ERISA, an EGFR inhibitor, the cells will die. But before they die, we can map out, it takes a couple of days. This here we're looking at 3 hours after the addition of ERISA. And we can map out detailed pathways and substrates downstream of the receptor tyrosine kinase. And so here we're looking with a phosphotyrosine antibody and characterizing phosphotyrosine signaling. And when we did these studies, we were very surprised to find the following, and that is that in all of the cell lines that were dependent on a single receptor tyrosine kinase, so when you give a single kinase inhibitor and they die, an RTK inhibitor, we found this kind of network that was established, so that the oncogenic kinase controlled the activity of all the other receptor tyrosine kinases in the cell. And so when we give the drug, in this case, we see loss of phosphorylation at the activation loops of all of the other receptor tyrosine kinases. So somehow, in this case, MET is controlling the activity of all these other kinases. It set up some kind of a hierarchy. And this is likely responsible, the fact that the kinase can control these other enzymes likely explains a number of activities that the cancer cell needs to carry out to be successful, such as metastasis. And so we think this is a hallmark of patients as well who are going to respond to single agent therapies. That is, this explains why a single agent therapy can work so well for RTK inhibitors, because you're essentially shutting off all of the receptor tyrosine kinases in the cell. So we've looked in a number of cell lines, and as I said, this seems to be a very common phenomenon. And here's 3 different cell lines driven by 3 different receptor tyrosine kinases. And again, we find the same kind of network that's established. We found other cell lines that are addicted to actually where we have 2 of these different kinds of networks present. For example, ALK is active and EGFR are active, and there are 2 separate networks. There, you need to give 2 separate drugs to get killing. So we think this could be an indication of the combination therapies that could be successful on patients. So we can look at, and I'm just showing here the RTK networks. Through this analysis, we find hundreds, or not thousands, of other downstream substrates, which I'm not showing here. But we can also look now with this, the activation loops in the serine threonine kinase space, and look at the kinases that are inhibited on their activation loop when we give the drug. And so now we can map the signaling downstream into the serine threonine kinases, and find lots of enzymes that are activated, that are controlled by the oncogenic kinase. Many of these, are well known, like AKT and ERK, and others are more novel, and these could represent, I think, important new drug targets downstream of the RTK. So we can look even further now downstream of the serine threonine kinases at their substrates. And this is a study that we published in Science Signaling. Al Moretz was the lead author on this, where we were using now an antibody that recognizes substrates of AKT-like enzymes, where there's a conservation of arginine at -5 and -3. We made an antibody that recognizes that motif. And we can identify many, many different substrates that are turned off when we give the RTK inhibitors. So we can establish these complex networks downstream of the RTK at multiple levels. And here we could identify lots of interesting new pathways. But I think one of the more interesting observations with regard to cancer comes back to the receptor tyrosine kinases. And this is, again, coming back to this cell line that's driven by c-Met. Met is obviously important in metastasis and motility. And what we could show is that when we inhibit Met, we also inhibit the phosphorylation of efferent A2 at a site that's critical for its motility. And this occurs at the level of AKT signaling. So there's multiple levels of coupling of connections between these receptor tyrosine kinases. At the tyrosine level, where Met is somehow leading to phosphorylation of the activation loops of the tyrosine kinase, other tyrosine kinases, receptor tyrosine kinases, and also now at the AKT level. So this is, I think, new and interesting connections that can be established that can explain how Met can subvert some of these other cellular activities for its use in cancer cells. Okay, so I've just described now how we can kind of map signaling at the tyrosine level and serine threonine kinase level. What about acetylation? Well, I'll tell you one quick story here. Here we've again surveyed broadly across all of the proteins whose acetylation changes when we give these RTK inhibitors. And the protein that changes the most is, probably not surprisingly, is c-Myc. And we found that acetylation at lysine 163 is strongly repressed when we give these receptor tyrosine kinase inhibitors. And this is a site that's well known to be important for stabilizing c-Myc and for promoting its transcriptional activation properties and increasing presumably the proliferative pathways. What was also known about this site is that it's acetylated by P300 and CBP, and when they interact with c-Mycs, they interact through this KIX domain. And when we looked at the KIX domain, we found a number of different tyrosine phosphorylation sites. So this immediately suggests that the RTKs could be regulating MYC activity through phosphorylation in some way of CBP, and this is something we could easily test. So we can analyze signaling at these multiple levels and I think uncover new and interesting regulatory phenomenons. So now let me move to the final vignette here, which is our work that we've been doing over the last 5 years or so to characterize signaling in tumors using this approach. And we have a number of scientists who have traveled over to China, made multiple trips over there. There's 3 of them are shown here, to acquire tumor tissues, which we processed in China and brought back as peptides. So we processed the tissues. And this just shows some of the samples that we've analyzed so far. In red are the tumors, in blue are the normal cell lines, and in green are the normal tissue, and in green are the cell lines. So we've analyzed almost 500 tumors in the tyrosine signaling space. And I'm going to talk to you mostly about signaling, the tyrosine data that we've identified. The basic idea is to profile the tumor tissue, profile the normal tissue, subtract the normal signaling that we see from the tumor, and then be left with a tumor-specific profile. And we can identify proteins that are activated in the tumor and in different types of tumors, and look at those together to form pathways and try to identify disease drivers. So let me show you a smattering of this kind of data. So here we're looking at 500 different patients across the top in the horizontal direction. And then in the vertical direction, we're looking at the 90 or so different tyrosine kinases. And this is, we're doing hierarchical clustering here, where we've already subtracted away the normal tyrosine signaling from the tumor. So this is tumor-specific signaling. And we're clustering patients based on the similarity of their tyrosine kinase profiles and tyrosine kinase activity. And I think there are 3 major features that I just wanted to point out in the data here. There's the activation of SRC, FAC, and DDR1. And when we look at each of these, like DDR1, we see that the patients aren't clustering according to tumor type, but rather according to the expression of the different kinase activities. So DDR1 will have a whole collection of different types of tumors that are want to activate this enzyme. So we could ask, why do tumors want to activate these different enzymes? FAC and SRC are pretty well described as playing important roles downstream of other receptor tyrosine kinases and other receptors in cancer. Whereas DDR1 is much less understood to be an important oncogene in cancer, or driver. So this kind of tweaked our interest, and we were asking, could DDR1 be a driver in cancer? And what's known about it? Well, it's known to be a receptor for collagen. It's overexpressed in many epithelial cancers, occasionally mutated in a few cancers. And the signaling is not really, downstream signaling is not well understood, but there is a few reports on its coupling to Notch. And so what we were able to do is to take the data that we had across the 500 tumors and try to assemble an associated network. So we asked, when DDR1 phosphorylation is high, when the activity of DDR1 is high, what other substrates are also high? And when DDR1 is low, what other substrates are also low? And so we could construct a network, an associated network in this way. And we worked together with Andre Califano at Columbia University, and we used a Gene Neighbors program from the Broad to construct this network. And the bottom line is, when we look at this network, it looks very much like an oncogenic network, very much like what we'd expect to see for EGFR, for example. So the strong inputs into PI3 kinase and AKT, metabolic activity, other tyrosine kinases, cytoplasmic tyrosine kinases, the MAP kinase pathway. So this looks very much like an oncogene, and this is something we really want to investigate further, and we're actually quite interested in making antibodies that might have therapeutic value here. Okay, so we can also look at the data and look at these clusters more carefully, and we see lots of other known oncogenes clustering together. But perhaps the best way to, I think, look at this data was to rank it based on the kinases that are most active in a given tissue. So here, we've constructed and ranked the kinases that we found to be most activated on a per sample basis. And when we look at what comes to the top, it's c-KIT, FGFR2, ALK, ROS, and then you find EGFR way down at the bottom here. I think fourth from the bottom is EGFR, which is clearly a known driver in many different tumors. So we think that all of these other enzymes above it are going to be important in the disease process, and that they need to be further investigated. But I'm just going to tell you a little story, and Mike mentioned this earlier, about two of these enzymes, ALK and ROS. And we were interested in these because they weren't normally expressed in epithelial cells, yet they were showing very high levels of activity in a number of tumor samples, actually a fairly small number, but they were the lowest hanging fruit that we investigated. So here they are on the kinase tree. ALK and ROS are very similar enzymes based on sequence analysis, and they're similar to IGF-1R and insulin receptor, and also DDR1, DDR2. They're in the same kinase branch. And so we investigated these further, and were able to show actually that ALK and ROS in lung cancer patients were found as gene fusions. And more recently, we found other gene fusions in ovarian cancer and liver cancer. And I'll tell you a little bit about this now. So this is the work that Mike mentioned, and the lead author is Clarissa Rakova, who's here in the audience today. So what we found was, back in 2007, this was published in Cell, that ALK was actually fused to EML4 and also this other protein, TFG. We found this in cell lines and in tumor samples. And we also found that ROS was fused to two other proteins in cell lines and in tumors. And that ALK was present as a fusion in about 4 or 5% of the Chinese patients, whereas ROS was present somewhere between 1 and 2% of the patients. And shortly before our paper came out, another paper came out in Nature from the Japanese group. And this piqued the interest of Pfizer, who was testing crizotinib, which had been developed through Sugen, as a MET inhibitor. And patients were, I think, on clinical trials for its activity on MET, when it was also noticed that it had off activity on ALK. And in fact, the patients who responded to this drug all had the EML4 ALK translocations. And so a larger scale phase 1 study was initiated. And it included only patients who had the EML4 ALK translocation, and was very, very successful. And this led, I think they skipped the phase 2 and went right to a phase 3 study, and it was approved in record time. And the reason this was so successful is because the only patients who were included in the clinical trial all had the EML4 ALK translocations. So this is, I think, a great story about finding the right drug for the right patient. And so we have been further studying these tumor samples and have found, and this is work that was published recently by Hong Ren, we have found ALK fusions also in ovarian cancer, this fibronectin fusion. And Hong has also found a number of patients who have wild type, what appears to be wild type ALK expressed in the serous subtype. And we could show that these fusions, we could create xenograft models, and that they were very sensitive to ALK inhibitors. So the bottom line is we think there's going to be a group of patients out here who will respond very well to Xalkori, which is now approved, who have these forms of ovarian cancer. We also found other ROS fusions in a liver cancer called cholangiocarcinoma. This is a very nasty disease. And we found Fig-ROS fusions, and this is work from Tingley Gu. We found Fig-ROS fusions in about 10% of these patients. And we could show that these fusion constructs are also strongly inhibited by ALK inhibitors. And that's what this central part of the graph is showing, the sensitivity to TAE, which is an ALK inhibitor, and they're equally sensitive to crizotinib. So the bottom line is we think that these patients will also respond well to crizotinib, Xalkori as it's now called. And very recently, there was a publication in the Journal of Clinical Oncology showing the first response, which was a complete response to one of the lung cancer patients with ROS fusion. So, and the rumors have it that these ROS patients are actually responding better to the ALK inhibitors than the ALK patients. So they seem to be responding very well. So to sum up, that ALK and ROS seem to be turning up here and there. We think that there could be up to 25,000 new patients a year who are going to have this, who could be candidates for treatment with this, with Xalkori. And we really need to get out and find those patients. And Herbert Hack is leading our efforts to develop antibodies that we can use in IHC to identify these patients and get them the medicines that they need, that will respond to. So I wanted to just come back for just a minute, tell you a little bit about some of the other areas we're looking at. So I just told you the story about ALK and ROS. What about the other serine threonine kinases, or the other levels of analysis that we can do on these tumors in acetylation and methylation, ubiquination? We're busily working on that, and with the goal of trying to find other drug targets and trace the signaling pathways further downstream into these other areas. And this just shows the analysis of about 90 different ovarian tumors, where we've reduced all the signaling in one space to a simple number, and we're looking at the difference in that signaling over normal tissue. And so what we can show here is that there are groups of patients who have very high tyrosine kinase activity, some groups of patients who have very high serine threonine kinase activity, patients who have high levels of acetylation or methylation. And now, since we have this massive amount of data, we can look in great detail to see what that activity means and trace it to the activated pathways. And so this is something we're actively pursuing to try to find new targets. So I just wanted to end with the idea that the next era of cancer therapeutics really requires looking at the cells and having the kinds of technologies that are going to allow us to understand what pathways are deregulated and put that into the context of the genetics. And I think we should all think back to the patient with that had the melanoma response, but relapse. We're clearly going to need a combination therapy, single agent therapies with advanced disease are not going to be sufficient. So we really need to understand these pathways at a level that will allow us to produce multiple drugs to hit the pathway at multiple points. So I'll end just by acknowledging the people who have contributed to the work. Clarissa Rakova, Aileen Gao, Ting Lei played a major role in a lot of what I described, as well as others listed here. Yi Wang in the bioinformatics, Peter Hornbeck, who leads our phosphocyte database, Kim Lee, Sean Beausoleil, and Jeff are all driving our mass spectrometry forward. And we work together with Steve Gigi's lab, who keeps us at the cutting edge of mass spectrometry. And I'd like to thank our collaborators in China for the patient samples and there.
For 2 questions, please.
So over here. So Mike, very nice talk. I have a question on the DDR data. So
Yeah.
did you have genomics on those patients, and were you able to identify a signature?
No, that would be great. I mean, these patients, because we can't transfer genetic material from China,
Right.
so all we have are peptides.
So you can't make the DNA at the same time. No. So just parenthetically, I would comment that earlier this year, I think, actually, we published as part of a large consortium in Toronto the results of a dropout screen of about 100 cancers, and 50% of them dropped out DDR1. So it would be very interesting to follow up on this.
Yeah, I think it could be important.
It's a strikingly similar number.
Any other questions? So Mike, you showed quite a few clustering situations where you have either where it's a signaling that actually drives the clusters. I'm wondering, have you tried to take it beyond clustering to try to actually predict, for instance, patient response or patient outcome from the network, which I think would be probably even more powerful, at least from a therapeutic point of view?
Yeah, we've thought about, what we'd like to be able to do is to understand, look at the tyrosine data and understand where the drivers are. And because these tumors are expressing multiple kinases, it's hard to look at that data and predict which is the driver and which is, controlled by that. So we need, I think, some new thinking and new approaches to help us.
Okay, well, let's thank Mike again.